Functions of large intestine. Learning objectives include list the composition of large intestinal secretion, list the functions of large intestine in relation to water and electrolyte absorption and gut fermentation. Next, glossary of terms. Crypts of Liberkin. Invaginations of the epithelium into the submucosa and contain specialized secretory cells. They are found in the small intestine and in the large intestine. Enteritis, inflammation of the intestine, is the enteritis. Secretion of mucus by the large intestine. Mucus secretion. The mucosa of the large intestine, like that of the small intestine, has many crypts of Liberkin. However, unlike the small intestine, it contains no villi. The epithelial cells secrete almost no digestive enzymes. Instead, they contain mucus cells that secrete only mucus. This mucus contains moderate amount of bicarbonate ions secreted by a few non-mucus secreting epithelial cells. The rate of secretion of mucus is regulated principally by direct tactile stimulation of the epithelial cells lining the large intestine and by local nerves reflexes to the mucus cells in the crypts of Liberkin. Stimulation of the pelvic nerves from the spinal cord, which carry parasympathetic innervation into the distal 1/2 to 2/3 of the large intestine, also can cause marked increase in mucus secretion. This increase occurs along with the increase in peristaltic movement of the colon, which is discussed after some time. Then during extreme parasympathetic stimulation, often caused by the emotional disturbances, so much mucus can occasionally be secreted into the large intestine that the person has a bowel movement of ropey mucus as often as every 30 minutes is mucus which contains little or no fecal material. So, large intestine is responsible for the mucus secretion when the person is emotionally, disturbed. So, composition of the large intestinal secretion. The large intestine secretion contain the following: water, mucus, Na plus Cl minus, bicarbonate, K plus Ca2 plus. So, when we just look at the structure of a crypts of Liberkin, found in all parts of the small intestine and large intestine between the villi, which secretes almost pure extracellular fluid. You can say that mucus goblet cells are there, epithelial cells and pinnate cells are there. Then functions of the mucus secretion. Mucus in the large intestine protects the intestinal wall against excoriation. It provides an adherent medium for holding fecal matter together. It protects the intestinal wall from the great amount of bacterial activity that takes place inside the feces. The mucus plus the alkalinity of the secretion, pH of 8.0, caused by the large amount of sodium bicarbonate, provides a barrier to keep acids formed in the feces from attacking the intestinal wall. Diarrhea causes by the excess secretion of water and electrolytes in response to irritation. Whenever a segment of the large intestine becomes intensely irritated, it occurs when bacterial infection becomes rampant during enteritis. The mucosa secretes extra large amount of water and electrolytes in addition to the normal viscid alkaline mucus. This secretion acts to dilute the irritating factors and to cause rapid movement of the feces toward the anus. This result is a diarrhea with loss of large quantities of water and electrolytes. However, the diarrhea also washes away irritant factors, which promotes earlier recovery from the disease than might otherwise occur. Extreme secretions of chloride ions, sodium ions, and water from the large intestinal epithelium in some types of diarrhea. Immature epithelial cells that continuously divide to form new epithelial cells are found deep in the spaces between the intestinal epithelial folds. These new epithelial cells spread outward over the luminous surface of the intestines. While still in the deep folds, the epithelial cells secrete sodium chloride and water into the intestinal lumen. This secretion in turn is reabsorbed by the older epithelial cells outside the folds, thus providing flow of water for absorbing intestinal digestate. The toxins of the cholera and some other types of diarrheal bacteria can stimulate the epithelial fold secretion so greatly that this secretion often becomes much greater than can be reabsorbed, thus sometimes causing a loss of 5 to 10 liters of water and sodium chloride as diarrhea each day. Within 1 to 5 days, many severely affected patients die for this loss of fluid alone. Extreme diarrheal secretion is initiated by entry of a subunit of cholera toxin into the epithelial cells. This subunit stimulates formation of excess cyclic adenosine monophosphate, which opens tremendous numbers of chloride channels, allowing chloride ions to flow rapidly from inside the cell into the intestinal crypts. In turn, this action is believed to be to activate the sodium pump that pumps sodium ions into the crypts to go along with the chloride ions. Finally, all this extra sodium chloride causes extreme osmosis of water from the blood, thus providing rapid flow of fluid along with the salts. All this excess fluid washes away most of the bacteria and is of value in combating this disease. But too much of a good thing can be lethal because of serious dehydration of a whole body that might ensure. In most instances, the life of a person with cholera can be saved by the administration of tremendous amounts of sodium chloride solution to make up for the loss. Absorption in the large intestine, formation of feces. About 1500 ml of chyme normally passes through the ileocecal valve into the large intestine each day. Most of the water and electrolytes in this chyme are absorbed in the colon, usually leaving less than 100 ml of fluid to be excreted in the feces. Also, essentially all the ions are absorbed, leaving only 1 to 5 equivalent each of sodium and chloride ions to be lost in the feces. Most of the absorption in the large intestine occurs in the proximal one half of the colon, giving this portion the name absorbing colon, whereas the distal colon functions principally for feces storage until a propitious time for feces excretion and is therefore called the storage colon. Absorption and secretion of electrolytes and water. The mucosa of a large intestine, like that of the small intestine, has a high capacity and capability for active absorption of sodium, and the electrical potential gradient created by sodium absorption causes chloride absorption as well. The tight junctions between the epithelial cells of the large intestinal epithelium are much tighter than those of the small intestine. This characteristic prevents significant amount of back diffusion of ions through these junctions, thus allowing the large intestine mucosa to absorb sodium ions far more completely, that is against a much higher concentration gradient than that can occur in the small intestine. This is especially true when large quantities of aldosterone is available because aldosterone gradually enhances sodium transport capability. In addition, as occurs in the distal portion of the small intestine, the mucosa of the large intestine secretes bicarbonate ions, while it simultaneously absorbs an equal number of chloride ions in the exchange transport process already described. The bicarbonate helps neutralize the acidic end products of the bacterial action in the large intestine. Absorption of sodium and chloride ions creates an osmotic gradient across the large intestinal mucosa, which in turn causes absorption of water. Maximum absorption capacity of the large intestine. The large intestine can absorb a maximum of 5 to 8 L of fluid and electrolytes each day. When the total quality entering the large intestine through the ileocecal valve or by way of large intestine secretion exceeds this amount, the excess appears in the feces as diarrhea. As noted earlier, toxins from the cholera or certain other bacterial infections often cause the crypts in the terminal ileum and large intestine to secrete 10 or more liters of fluid each day, leading to severe and sometimes lethal diarrhea. Bacterial action in the colon. Numerous bacteria, especially colon bacilli, are present even normally in the absorbing colon. They are capable of digesting small amounts of cellulose, in this way providing a few calories of extra nutrition for the body. In herbivorous animals, the source of energy is sufficient, although it is of negligible importance in human beings. Other substances formed as a result of bacterial activity are vitamin K, vitamin B12, thiamine, riboflavin, and various gases that contribute to flatus in the colon, especially carbon dioxide, hydrogen gas, and methane. The bacteria formed of vitamin K is especially important because of the amount of this vitamin in the daily ingested food is normally insufficient to maintain adequate blood coagulation. Fermentation in the large intestine. Fermentation is an important process that occurs in the large intestine. It is a process where bacteria, mainly anaerobic, and yeasts break down dietary substrates and other substrates present in the large intestine to obtain energy for their normal growth and maintenance of cellular functions. Substrates for fermentation. Many substrates are available for fermentation in the large intestine from dietary and endogenous sources. However, the most important ones are starch, dietary fiber, non-starch polysaccharides, and protein. Composition of the feces. The feces normally are about 3/4 water and 1/4 solid matter, that is composed of about 30% dead bacteria, 10 to 20% fat, 10 to 20% inorganic matter, 2 to 3% protein, and 30% undigested rock from the food and dried constituents of digestive juices such as bile pigment and slaughtered epithelial cells. The brown color of feces is caused by stercobilin and urobilin, derivatives of bilirubin. The odor is caused principally by products of bacterial action. These products vary from one person to another depending on their person's organic bacterial flora and on the type of food eaten. The actual odoriferous products include indole, skatole, mercaptans, and hydrogen sulfide. So there are many process of gut fermentation happening. Substrates in the colon like carbohydrates, proteins, and others. Bacterial and yeast enzymes break down substrates. Then breakdown products are like short chain fatty acids, acetic acid, propionic acid, butyric acid, branched chain fatty acids, ammonia, carboxylic acids, phenolic acids, amines. Then fate of the breakdown products are like absorbed, eliminated in feces, rectal gases utilized by microflora, SCFA, butyrate, primary nutrient for colonocytes, etc. Then probable substrates for fermentation are carbohydrate like starch, non-starch polysaccharides, dietary fiber, pectin, cellulose, hemicellulose, unabsorbed sugars, modified cellulose, polydextrose, etc. Protein, dietary proteins, endogenous proteins such as digestive enzymes, then others, intestinal glycoproteins and mucopolysaccharides. Then summary of the functions of the large intestine. Reabsorption of water and maintenance of fluid and electrolyte balance helps in the formation of stools, facilitates fermentation processes, absorption of certain end products of fermentation such as butyrate, vitamin B12, vitamin K, thiamine, and riboflavin, storage of fecal matter until eliminated. Next major topic that we are going to learn about is the gastrointestinal motility. Our learning objectives include list the different types of intestinal movements, what is the law of the gut, explain the stages and mechanism of deglutition, and list the factors influencing gastric emptying, list the stages of defecation and the factors regulating it. Before moving on to this complex topic, let's learn some major terms associated with this topic, like jargons of this topic. Chyme, food in the stomach that has been thoroughly mixed with the stomach secretions and passed down the gut. Then deglutition, the act of taking solids and liquids into the gastrointestinal tract through the mouth and throat. Mastication, the process of chewing and grinding food in the mouth. The time that food remains in each part of the alimentary tract is critical for optimal processing and absorption of nutrients. In addition, appropriate mixing must be provided because the requirements for mixing and propulsion are quite different in each stage of processing. Multiple automatic nerves and hormonal mechanisms control the timing of each of these activities, so they will not occur optimally, not too rapidly and not too slowly. This chapter discusses its movements, especially the automatic mechanisms of this control. So, functional types of movements in the gastrointestinal tract. Two types of movements occur in the gastrointestinal tract: propulsive movements and mixing movements. Propulsive movements, which cause food to move forward along the tract at an appropriate rate to accommodate digestion and absorption. Which cause food to move forward along the tract at an appropriate rate to accommodate digestion and absorption. So, mixing movements, which keep the intestinal contents thoroughly mixed at all times. So, mixing movements, which keep the intestinal contents mixed at all times. First is the propulsive movements or peristalsis. The basic propulsive movement of the GI tract is peristalsis. A contractile ring appears around the gut and then moves forward. This mechanism is analogous to putting one's fingers around a thin, distended tube and then constricting the fingers and sliding them toward along the tube. Any material in front of the contractile ring is moved forward. Peristalsis is an inherent property of many syncytial smooth muscle tubes. It occurs in the bile ducts, glandular ducts, ureters, and many other smooth muscle tubes of the body. The usual stimulus for intestine peristalsis is distension of the gut. Other stimuli that can initiate peristalsis include chemical or physical irritation of the epithelial lining in the gut. Also, strong peristaltic nerve signals to the gut will elicit strong peristalsis. Function of the myenteric plexus in peristalsis. Peristalsis occurs only weakly or not at all in any portion of the GI tract that has congenital absence of the myenteric plexus. Also, it is greatly depressed or completely blocked in the entire gut when a person is treated with atropine to paralyze the cholinergic nerve endings of the myenteric plexus. Therefore, effectual peristalsis requires an active myenteric plexus. Peristaltic veins move toward the anus with downstream receptive relaxation law of the gut. Peristalsis theoretically can occur in either direction from a stimulated point, but it normally dies out rapidly in the towards the mouth direction while continuing for a considerable distance toward the anus. The exact cause of this direction transmission of peristalsis has never been ascertained, although it probably results mainly from the fact that the myenteric plexus is polarized in the anal direction, while it can be explained as follows. When a segment of intestinal tract is excited by the distension and thereby initiates peristalsis, the contractile ring causing the peristalsis normally begins on the orad side of the distended segment and then moves towards the distended segment, pushing the intestinal contents of the anal direction for 5 to 10 cm before dying out. At the same time, the gut sometimes relaxes several centimeters downstream toward the anus, which is called deceptive relaxation, thus allowing the food to be propelled more easily towards the anus than towards the mouth. This complex pattern does not occur in the absence of a myenteric plexus, therefore the complex is called the myenteric reflex or the peristaltic reflex. The peristaltic reflex plus the anal direction of movement of the peristalsis is called the law of the gut. Next, mixing movements. Mixing movements differ in different parts of the alimentary tract. In some areas, the peristaltic contractions cause most of the mixing. This is especially true when forward progression of the intestinal contents is blocked by a sphincter, so that a peristaltic wave can then only churn the intestinal contents rather than propelling them forward. At other times, local, intermittent, constrictive contractions occur every few centimeters in the gut wall. These contractions promote the chopping and shearing of the contents. Then, ingestion of food. The amount of food that a person ingests is determined principally by an intrinsic desire for food called hunger. The type of food that a person preferentially seeks is determined by appetite. The correct discussion is confined to the mechanics of food digestion and ingestion, especially mastication and swallowing. Then peristaltic contraction, leading wave of distension, 0 time, 5 seconds later what is happening. So then mastication or chewing. Chewing is an important for digestion of all foods, but especially important for most fruits and raw vegetables because these have indigestible cellulose membrane around their nutrient portions that must be broken before food can be digested. Also, chewing aids in the digestion of food for still another simple reason. Digestive enzymes act only on the surface of food particles, therefore the rate of digestion is absolutely dependent on the total surface area exposed to the digestive secretions. In addition, grinding the food to a very fine particulate consistency prevents excoriation of the GI tract and increases the gauge with which food is emptied from the stomach into the small intestine and then to all the succeeding segments of the gut. Most of the muscles of the chewing are innervated by the motor branch of the fifth cranial nerve, and the chewing process is controlled by nuclei in the brainstem. Stimulation of specific reticular areas in the brainstem taste centers will cause rhythmic chewing movements. Also, stimulation of areas in the hypothalamus, amygdala, and even the cerebral cortex near the sensory areas for taste and smell can often cause chewing. Then chewing reflex. Much of the chewing process is caused by a chewing reflex. Presence of a bolus of food in the mouth, reflex inhibition of the muscles of mastication. Lower jaw drops initiates a stretch reflex. Rebound contraction raises jaws, closure of teeth. Compresses the bolus again against the oral lining and this process of mastication repeats. Then swallowing, deglutition. Swallowing is a complicated mechanism, principally because the pharynx subserves respiration and swallowing. The pharynx is converted for only a few seconds at a time into a tract for propulsion of food. It is especially important that respiration not be compromised because of swallowing. Stages of deglutination have been very important, and then voluntary stage of swallowing. When the food is ready for swallowing, it is voluntarily squeezed or rolled posteriorly into the pharynx by pressure of the tongue upward and backward against the palate. From here on, swallowing becomes almost entirely automatic and ordinarily cannot be stopped. Then stages of deglutition: voluntary stage, pharyngeal stage, and esophageal stage. So voluntary stage initiates the swallowing process. Pharyngeal stage is involuntary and constitutes passage of food through the pharynx into the esophagus. Then esophageal stage is there, so another involuntary phase that transports food from the pharynx to the stomach region. Food from the pharynx to the stomach. So involuntary pharyngeal stage of swallowing. As the bolus of food enters the posterior mouth and pharynx, it stimulates epithelial swallowing receptor areas all around the opening of the pharynx, especially on the tonsillar pillars, and impulses from these areas pass to the down brainstem to initiate a series of automatic pharyngeal muscle contractions. Then involuntary pharyngeal stages of swallowing. As the bolus of food enters the posterior mouth and pharynx, it stimulates epithelial swallowing receptor areas all around the opening of the pharynx, especially on the tonsillar pillars, and impulses from these areas pass to the brainstem to initiate a series of automatic pharyngeal muscle contractions as follows. The soft palate is pulled upward to move the posterior narrows to prevent reflux of food into the nasal cavities. The palatopharyngeal folds on each side of the pharynx are pulled medially to form a sagittal slit through which the food must pass into the posterior pharynx. Because this stage lasts less than 1 second, any large object is usually impeded too much to pass into the esophagus. The vocal cords of the larynx are strongly approximated and the larynx is pulled upward and anteriorly by the neck muscles, and the epiglottis swings backward over the opening of the larynx. These actions prevent passage of food into the nose and trachea. Most essential is the tight approximation of the vocal cords, but the epiglottis helps to prevent food from never getting as far as the vocal cords. Destruction of the vocal cords or of the muscles that approximate them can cause strangulation. The upward movement of the larynx also pulls up and enlarges the opening of the esophagus. At the same time, the upper 3 to 4 cm of the esophageal muscular wall, called the upper esophageal sphincter, also called the pharyngoesophageal sphincter, relaxes. Thus, food moves easily and freely from the posterior pharynx into the upper esophagus. Between swallows, this sphincter promotes strongly contractor, thereby preventing air from getting into the esophagus during respiration. The upward movement of the larynx also lifts the glottis out of the main stream of food flow, so the food mainly passes on each side of the epiglottis rather over its surface. This action adds still another protection against the entry of food into the trachea. Once the larynx is raised and the upper esophageal sphincter becomes relaxed, the entire muscle wall of the pharynx contracts, beginning in the superior part of the pharynx and then spreading downwards over the middle and interior pharyngeal areas, which propels the food by peristalsis into the esophagus. To summarize the mechanisms of the pharyngeal stage of swallowing, the trachea is closed, the esophagus is opened, the fast peristaltic wave initiated by the nerve system of the pharynx forces the bolus of food into the upper esophagus. The entire process occurs in less than 2 seconds. Nervous initiation of the pharyngeal stage of swallowing. The most sensitive tactile areas of the posterior mouth and pharynx for initiating the pharyngeal stage of swallowing lie in a ring around the pharyngeal opening, with greatest sensitivity in the tonsillar pillars. Impulses are transmitted from these areas through the sensory portions of the trigeminal and glossopharyngeal nerves into the medulla oblongata, either into or closely associated with the tractus solitarius, which receives essentially all sensory impulses from the mouth. The successive stages of the swallowing process are then automatically initiated in orderly sequence by neuronal areas of the reticular substance of the medulla and lower portion of the pons. The areas in the medulla and lower pons that control swallowing are collectively called deglutination or swallowing center. The motor impulses from the swallowing center of the pharynx and upper esophagus that cause swallowing are transmitted successively by the 5th, 9th, and 10th and 12th cranial nerves, and even a few of their positive superior cervical nerves. In summary, the pharyngeal stage of swallowing is principally a reflex act. It is almost always initiated by voluntary movement of food into the back of the mouth, which in turn excites involuntary pharyngeal secretory receptors to elicit the swallowing reflex. Effect of the pharyngeal stage of swallowing on respiration. The entire pharyngeal stage of swallowing usually occurs in less than 6 seconds, thereby interrupting respiration for only a fraction of a usual respiratory cycle. The swallowing center specifically inhibits the respiratory center of the medulla during this time, halting respiration at any point in the cycle to allow swallowing to proceed. Yet, even while a person is talking, swallowing interrupts respiration for a short time, it is hardly noticeable. The esophageal stage of swallowing involves 2 types of peristalsis. The esophagus functions primarily to conduct food rapidly from the pharynx to the stomach, and its movements are organized specifically for this function. The esophagus normally exhibits 2 types of peristaltic movements, like primary peristalsis and secondary peristalsis. So, primary peristalsis is simply a continuation of the peristaltic wave that begins in the pharynx and spreads into the esophagus during the pharyngeal stage of swallowing. This wave passes all the way from the pharynx to the stomach in about 8 to 10 seconds. If the primary peristaltic wave fails to move all the way, that is, has entered the esophagus into the stomach, secondary peristaltic waves results from the disorientation. Distension of the esophagus itself by the retained food. This waves continue until all the food has emptied into the stomach. The secondary peristaltic waves are initiated partly by intrinsic neural circuits in the meninteric nerve system and partly by reflexes that begin in the pharynx and are then transmitted upward through vagal afferent fibers to the medulla and back again to the esophagus through gasopharyngeal and vagal afferent nerve fibers. The musculature of the pharyngeal wall and upper third of the esophagus is striated muscle. Therefore, the peristaltic waves in these regions are controlled by skeletal nerve impulses from the glossopharyngeal and vagus nerve. In the lower two-thirds of the esophagus, the musculature is smooth muscle, but this portion of the esophagus is also strongly controlled by the vagus nerves that acts to the connections with the esophageal meninteric nerve system. The receptive relaxation of the stomach. When the esophageal peristaltic wave approaches the stomach, a wave of relaxation transmitted through meninteric inhibitory neurons precedes and peristalsis. Furthermore, the entire stomach and to a lesser extent even the duodenum becomes relaxed as this wave reaches the lower end of the esophagus and thus are prepared ahead of time to receive the time propelled into the esophagus during the swallowing act. Then functions of the lower esophageal sphincter. Gasoesophageal sphincter. At the lower end of the esophagus, extending upward about 3 cm above its juncture and the stomach is the broad lower esophageal sphincter, also called the gastroesophageal sphincter. The sphincter, consisting of circular muscle, normally remains tonically constricted with an intraluminal pressure of about 30 mmHg, in contrast to the mid portion of the esophagus, which normally remains relaxed. When the peristaltic swallowing wave passes down the esophagus, the receptive relaxation of the lower esophageal sphincter occurs ahead of the peristaltic wave, which allows easy propulsion of the swallowing food into the stomach. Rarely, the sphincter does not relax satisfactorily, resulting in a condition called achalasia. The tonic contraction of the lower esophageal sphincter helps prevent significant reflux of acid stomach contents into the esophageal content except under abnormal conditions. Additional prevention of esophageal reflux by valve-like closure of the distal end of the esophagus. Another factor that helps prevent reflux is a valve that, like a mechanism of a short portion of the esophagus that extends slightly into the stomach. Increased intra-abdominal pressure caves the esophagus inward at this point. This valve-like closure of the lower esophagus helps to prevent high intra-abdominal pressure from facing stomach contents backward into the esophagus. Otherwise, every time we walked, coughed, or breathed hard, we might expel stomach acid into the esophagus. Motor functions of the stomach. The motor functions of the stomach are threefold: storage of the large quantities of food until the food can be processed in the stomach, duodenum, and lower intestinal tract. Then mixing of these food with gastric secretions until it forms a semi-fluid mixture called chyme. Slow emptying of the chyme from the stomach into the small intestine at a rate suitable for proper digestion and absorption by the small intestine. Basic anatomy of the stomach can be seen like esophagus, cardia region, fundus region, body, rugae region, then pyloric sphincter, antrum, angular notch, pylorus, and duodenum. That is the physiological anatomy of the stomach. Anatomically, the stomach is usually divided into two major parts, the body and the antrum. Physiologically, it is more appropriately divided into a orad portion comprising about the first 2/3 of the body and the caudad portion. Orad portion and caudad portion comprising the remainder of the body plus the antrum. Next, storage function of the stomach. As food enters the stomach, it forms concentric circles of the food in the orad portion of the stomach, while the newest food lying close to the esophageal opening and the oldest food lying near the outer wall of the stomach. Normally, when food stretches the stomach, a vagovagal reflex from the stomach to the brainstem and then back to the stomach reduces the tone of the muscular wall of the body of the stomach so that the wall bulges progressively outward, accommodating greater and greater quantities of food up to a limit in the completely relaxed stomach of 0.8 to 1.5 liter. The pressure in the stomach remains low until this limit is approached. Mixed and propulsion of food in the stomach. Basic electrical rhythm of the stomach wall. As long as food is in the stomach, weak peristaltic constrictor waves called mixing waves begin in the mid to upper portions of the stomach wall and move toward the antrum about every 15 to 20 seconds. These waves are initiated by a gut wall basic electrical rhythm consisting of electrical slow waves that occur spontaneously in the stomach wall. As the constrictor waves progress from the body of the stomach into the antrum, they become more intense. Some become extremely intense and provide powerful peristaltic action potential, drive in constrictor rings that force the antral contents under higher and higher pressure towards the pylorus. These constrictor rings also play an important role in mixing the stomach contents by digging deeply into the food contents in the antrum. During gastric motility, most of the antral contents are squeezed upstream through the peristaltic ring toward the body of the stomach, not through the pylorus. Thus, the moving peristaltic constrictor ring, combined with its upstream squeezing action, called retropulsion, is an exceedingly important mixing mechanism in the stomach. After food in the stomach has become thoroughly mixed with the stomach secretions, the resulting mixture that passes down to the gut is called chyme. The appearance of chyme is that of a murky semi-fluid or paste.